Okay, students, you're welcome back to genetics series. We continue our study of genetics as applied to animal breeding for the improvement of animals. Today we're looking at module 18. Here we'll be talking about semen and embryo sex. So attempts have been made at developing techniques for the separation of X and Y bearing spermatozoa. The use of sex semen is now very common, especially in the dairy industry, where the desire is to produce female offspring. This may also be applicable in the poultry industry, especially where layers are being produced, and this will also help reduce the issue of the production of cockerels, because eggs can be sexed so that only the females can be produced. Now, semen can be sorted into X and Y bearing ones through a method or a technique known as flow cytometry. And through this method, the X and Y bearing sperm cells are separated, packaged for further use. The success rate has been put at around 90 to 95%, which is quite high and very good. So, real and apparent differences that exist between X and Y bearing sperm chromosome is used to separate them. Now, what are those differences? There's difference in the mass, the surface charge, antigenic property, and the buoyant density of the two sperm cells. And is it the most reliable method? The most reliable method is to utilize the slight differences in the DNA content of the X and Y bearing sperm cells. Through this method, these slight size differences between the X and Y sperm bearing chromosome is easily detected. The technique stains the sperm cells with fluorescent stains. The difference is related to the degree of fluorescence, and the X and Y bearing sperm cells pass through laser beams, which are used for identification purposes. So this is the process of flow cytometry, where it is used in separating X and Y sperm bearing cells. Excuse me. So the procedure as follows. Sperm cells are dyed with DNA dye, and the X bearing sperm cells bind more to the dye than the Y bearing sperm cells. So only one sperm cell is released in a drop. The drop is through this tube here, and as it goes through the tube, it gets to this point, laser beams excite the dye. The sperm cells then gives us light that is proportionate or proportional to the DNA content. So the more the light that is emitted, the greater the amount of DNA in the sperm cell and vice versa. The drops become charged at this point depending on the light intensity. The X sperm cell emits more light. And then as it passes through here, the drop passes through a pair of electrodes, positive and negative, and the charge drops are sorted into different tubes. Those that are attracted to the positive are the X bearing sperm cells, and those that are attracted to the negative are the Y bearing sperm cells. So once these have been collected, animals can then be inseminated using the sperm cells. As sperms are sexed, so we can do embryo sexing, and this is used in regulating animal gender. And embryo sexing is based on the removal of a small number of cells from the embryo at the 16 cell stage. or it may be done at later stage of development. So there are two embryo sexing procedures. We have what is known as the intrusive or the invasive, and the non-intrusive or non-invasive embryo sexing procedures. The non-intrusive one is deemed to be the best, and this is because the embryo integrity is preserved. There is also increase in the ability for normal development. However, it's less accurate when compared to the intrusive procedures. So, what is the importance of embryo sexing? It is used to predict an animal's sex. It is used for the diagnosis of genetic disorders in utero. It is beneficial for animal breeding and management. It is an option to run fewer recipient females while rapidly growing your head size. Embryo transfer will benefit greatly from this method, and it is used to protect threatened and endangered animal species, and it is easier to pre-select animal gender using embryo sexing. So there are many ways of sexing embryos. Let's take a look at the invasive methods. And the first one is the use of karyotyping, or what is also known as cytogenic analysis. And this involves collecting individual cells, culturing them, and then analyzing the chromosomes under a microscope. And the process identifying the sex chromosome as either an X or a Y chromosome. However, there are problems attached to this procedure, and these are getting the biopsies with high quality metaphase chromosome spreads. There is a potential harm to the developing embryos. It is labor intensive, time demanding, and requires even up to about 5 hours. And there's also the need for skilled cytogenists, at least you need two to handle up to 50 embryos at a time. It requires a large number of cells, and there's usually the difficulty of obtaining sufficient cells for analysis, especially at the early stage of the embryo growth. And the success rate seldom reaches 62% because of weak metaphase chromosome spread. So these are the problems that are linked to karyotyping. The second one to look at is embryo sexing using what is known as V-specific DNA probes. Again, the procedure is as follows. The zona pellucida breach that broken into the embryo, and a single cell is extracted at the 6 to 10 cell stage 3 days after in vitro fertilization. So the Y-specific DNA is then amplified using PCR primers. PCR is polymerase chain reaction. It's in this process small parts of the 149 base pair D chromosome specific probe is taken into consideration. Amplification of the DNA. After it has been done, the DNA is now electrophoresed and stained with a dye, ethidium bromide, and filled under ultraviolet light. So the embryo is identified as male if the 149 base pair fragment can be clearly seen after amplification. If it is not clearly seen, then the embryo is female. Then analysis of the amplified product is done, and then it is then interpreted. So what are the advantages of this method? It's very accurate. It could be done even during early pregnancy using DNA that is taken from the maternal plasma. Although it involves surgery, It is completed in very few hours. It is cost effective, simple, and very reliable. And this method necessitates, even though it necessitates the removal of cells from the embryo, PCR setting is nearly 100% correct. Then we have what is known as embryo sexing using fluorescent in situ hybridization of fish. This technique uses fluorescently labeled probes, and in the process it can identify or locate specific DNA sequences on chromosomes, therefore enabling sex determination. It involves a number of steps. The first one is embryo collection and preparation. So here embryos are collected from donor animals through uterine flushing or in vitro fertilization procedures. And the outer zone of the embryo, what is called the zona pellucida, is removed mechanically or enzymatically to allow for better access for the probe. These embryos are then separated into individual cells or flattened onto microscope slides. Then comes cell fixation, during which the separated embryonic cells are fixed into microscope slides using a fixation solution such as methanol to acetic acid. And this preservation or fixation preserves the cell structure and immobilizes the chromosomes, thereby making them accessible for hybridization. The next stage is the denaturization of the chromosomes. So during which the fixed chromosomes are denatured using heat or chemical solution. So why are we denaturing? So that we can separate the double-stranded DNA into single-stranded ones. Why do we need to have single-strand DNA? This is essential because the fish probes are single-stranded and need to bind to single-stranded target DNA. The third process is known as probe preparation and hybridization. During this time, sex-specific DNA probes are designed to target a particular sex chromosome, be it the X or the Y chromosome, and the probes are at this point labeled with different fluorescent dyes. These fluorescent dyes are called fluoroprobes. After that, we now carry out the hybridization, and during which time the fluorescently labeled probes are applied to the denatured chromosomes on the slide. They are incubated under controlled conditions of temperature and humidity. This allows the probe to hybridize, that is, allows it to bind to their complementary sequences on the chromosomes. Then comes washing. This is a critical step as it reduces background fluorescence and ensures accurate signal detection. During this process, slides are washed rigorously to remove any unbound or non-specifically bound probes. Then comes counterstaining, whereby a DNA counterstain, which is DAPI, which stains all DNA blue, is applied so that it can enable the visualization of the nuclei and the chromosomes, providing morphological content for the fluorescent signals. Then this is now viewed under the microscope, and here the slides are examined under a fluorescent microscope that is equipped with different filters for each fluorochrome. And each of the filters is able to act on each of the fluorochrome, and then we can now interpret the result such that the embryo is female when two fluorescent signals corresponding to the X chromosomes probes, e.g., we may have two red signals to be observed in each cell's nucleus, and it is male when only one fluorescent signal for the X chromosome probe, that is, e.g., one red signal and then one fluorescent signal for the Y chromosome probe, e.g. one green signal. So we can see that when the embryo is female, the signal will be the same color, and if it is male, the signal will be two different colors. So apart from embryo sexing, the fish process can also help in detecting aberrations in the chromosome in the embryo. For instance, it can detect what is known as mosaicism. And mosaicism is the presence of two or more cells lined with different genetic makeups, but within a single individual. And this comes about from a single fertilized egg. It can also detect what is known as aneuploidy. This is an abnormal number of chromosomes in a cell, while at the same time determining an embryo sex. Fish can also do these too. The lowdown of fish, however, is that it is very complicated. It requires technical know-how. It is expensive, time-consuming, and then there's a danger that the embryo ability may not be good after the process. However, it is 86.66% accurate, and therefore it is a good, and it has great applicability, especially when you are using small samples. So we come to the non-invasive methods, and the first one we're looking at the X-link, the use of X-link enzymes. So the concept of X-link enzyme is based on the phenomenon of X inactivation, or what is known as lionization in female mammals, and then the differential expression of X-linked genes between the sexes. So step-by-step explanation. First, we have to have an understanding of what X-linked genes and enzymes are. So X-linked genes are genes that are located on the X chromosome, and many of them code for certain enzymes. And we recall that a male have one X chromosome, while females have two X chromosomes. So X inactivation in females occurs during early embryonic development, whereby one of the two X chromosomes in each somatic cell, these are non-sex cells, is randomly inactivated. The inactivated X chromosome will condense thereafter into a compact structure called bar bodies, and this ensures that females, despite having two X chromosomes, do not produce twice the amount of X-linked genes or gene products compared to males who only have one X chromosome. So this is a form of dosage compensation. This is nature's way of preventing the overproduction of X-linked gene products from the X chromosome. The inactivation is however random. In some cells, it is the paternal or the father's X that is inactivated, while in others, it is the mother's X that is inactivated. So this leads to a mosaic pattern of expression in female for X-linked traits. Homozygosity and enzyme isoforms. So if a female embryo is heterozygous for an X-linked gene that code for an enzyme, this means that she has two different alleles for that gene. Then one allele will produce enzyme A, while the other will end up producing enzyme B. Then, due to X inactivation, some cells will end up expressing enzyme A, and the other cells will end up producing or expressing enzyme B. These different forms, it is these different forms of the same enzymes that are called isoforms. Isoforms. So what is the theoretical basis for sexing? The theoretical basis is such that female embryo carrying excess. In them, cells obtained from female embryo that is heterozygous for an X-linked enzyme, when analyzed, shows that the enzyme activity or the protein isoform will form or show the presence of both enzyme isoform, that is both enzyme A and enzyme B. And this is because some of the cells would have inactivated one X and expressed the other, so, and vice versa, leading to a mix of cells expressing different isoforms. So if cell A is inactivated, it produces a type of enzyme, while the other cell that is inactivated will produce the other type of enzyme. In male embryo XY, because the male only have one X chromosome, so it will only express one allele for any given X-linked gene. Cell analysis from the male embryo will therefore show only one enzyme isoform. It can either be an A or a B, depending on which allele is inherited from the mother. The next method of the non-intrusive one is the use of male HY antigen in SEXING embryos. So XY antigen is a male-specific protein that is expressed due to the presence of the Y chromosome. This can be taken advantage of for embryo sexing in animals. The step-by-step procedure shows that the first thing to do is to collect embryo and prepare it. Then this embryo is fixed. Even though it is optional, embryos are fixed on 4% paraformaldehyde. This takes for about 15-20 minutes at room temperature. It is then washed 3 times with PBS. PBS is phosphate sulfide solution, and it is used to remove debris. Stage 3, or is known as permeabilization, during which embryos are treated with 0.1 to 0.5 trecton X100, again in PBS for 10 to 15 minutes. Again, wash 3 times. Then comes blocking. This is done to reduce non-specific binding, and during this process, embryos are incubated in 1 to 5% BSA or bovine serum albumin in PBS for up to 60 minutes at room temperature. Then there's primary antibody incubation. So during this process, an anti-HY antibody is added to the embryo and then incubated for 1 to 2 hours at room temperature, overnight at 4 degrees. It's again washed thrice with PBS in order to remove any compound antibody. There's a secondary antibody incubation. During this, a fluorophore or enzyme conjugated secondary antibody could be fluorescein isocyanide labeled anti-mouse immunoglobulin G is added and incubated for 1 hour at room temperature in the dark. Again, washed thrice with PBS. Then the detection process. So when the embryo is observed under a fluorescent microscope, it is a male when the HY is positive, otherwise it is female when it is negative. So with enzyme-based method, a bright field microscope is used to detect color change. So it is male if there's color change, and it is female when there's no color change. To validate, there may be need to use PCR-based sexing. So this will help in making it more accurate, as the case may be. And when you are doing this, is by amplifying the Y chromosome specific genes like the SRY gene. So another process that can be used is hormonal assay, which is used for sexing blastocysts. This is another non-intrusive method. And here sexing of embryos is through the use of hormonal assays, which depends on the detection of sex-specific hormones, e.g. estrogen and testosterone. Estrogen is for the female and testosterone is in the male. That these hormones are produced by the embryo or its surrounding environment. So this method can be used in livestock breeding and in what is known as ART, assisted reproductive technologies. So the procedure as follows. One, you do embryo culture and hormone selection. So here embryos are cultured individually or it can be done in small groups in fresh medium. So the mediums for 24 to 48 hours at this temperature and at this percent carbon dioxide. So serum containing media should be avoided. This is to prevent any background hormone interference. So we are at hormone extraction using ELISA kit. So some ELISA kits are required for this process, hormone extraction. For example, using diethyether for steroids. And when using ELISA kit, it is best to follow the manufacturer's protocol step by step in order not to miss out on anything. The hormone is detected via ELISA. For example, we have ELISA kit for estradiol, which detects female embryos and testosterone for male embryos. Again, very important to follow the protocol. Then the data is analyzed, and this is done by comparing the absorbance level to a standard curve. So the result will show that female blastocysts will show higher estradiol levels, and the male will show higher testosterone levels. Then we come to sexing of embryo, which is based on cleavage and the developmental patterns. And this is possible because studies have suggested that the male and the female embryos exhibit differences in their cleavage timing, their morphology, and also their developmental rates. So these differences can be taken advantage of and used for primarily screening uh, in conjunction with other sexing techniques. For example, the use of hormonal assays or PCR-based methods. So what are the observed differences between male and female embryos? Look at cleavage speed, for example, which is faster in males, slower in females. The blastocyst formation takes place earlier in males and while it is slightly delayed in females. The cell number, it is higher in males. In males, you have higher inner cell mass cells, while in female, you have higher trophed, trophed cells. When you consider the morphology, male embryos are more compact and symmetrical, while female embryos are slightly irregular in shape. So these are the steps undertaken. First is to collect the embryo and culture it. So here we obtain zygotes or early embryos, for example at the 1 to 2 days post fertilization. They are cultured in a time-lapse incubator or observed at fixed intervals. So next, the cleavage timing analysis. The cleavage timing is the time taken to reach key stages which are monitored. The first cleavage is at the 2-cell stage. If you can get cleavage at this 2-cell stage, it means that the is a male, and because the males cleave faster. And If you get it at the second cleavage stage, the first cell stage, then this would likely be a female because they cleave at 1 to 2 hours later than the males. The blastocyst development assessment. So the blastocyst formation time can also be tracked. Males typically form blastocysts earlier. By day 5 to 6, they would have formed that, as observed in bovines, while the female may reach the same stage at 6 to 12 hours later. So another way is you can do, like we say, the morphological evaluation. So the blastocyst structure can be used to detect whether the embryo is male or female. And we say that the males have more compact inner cell mass that forms the embryo, that leads to the embryo having symmetrical shape, while the females have a larger trophoectoderm, but these are slightly irregular in morphology. So we can now carry out a statistical correlation, and this shows that embryo development will be compared against known sex ratios from where previous data already exist. For example, in cattle, uh, it's about 60% of fast growing cleavages are being observed to be males. So when we have this, we can be able to be almost certain of the sex on the embryo. However, if we are not 100% sure, we can validate, and this is through confirmation again using the PCR method where the SIY gene is amplified, or the hormonal assay method where we use hormones produced by the embryo, that is estradiol or testosterone, to know if it is a female or a male respectively. So students, we have come to the end of this module on sexing of sperm cells and the embryo. This has helped us in improving animals, particularly as we said at the beginning, where this particular sex of an animal is desired, as in the dairy industry and also in the egg laying industry. So take your time to like the channel, you can drop a comment, share your thoughts with others, or share this material with others, and please don't forget to subscribe. So we'll see you next module. Have a great day.